In this class, I will show you how our immune system actually reject the transplanted tissue or organ. But in the beginning, I will show you 3 techniques by which doctor or researcher actually come into conclusion that the transplantation should be performed or not. The first is ABO blood group antigen. The second is major histocompatibility complex. This amazing molecule in case of human is called human leukocyte antigen, HLA. And the third antigen that is responsible for this rejection is actually these 3 alloantigens are very important, are called minor histocompatibility antigen. So for these 3 alloantigen, intense rejection occur. Now in this 3 technique, what happens? Researcher or doctor actually sequence the HLA gene of the recipient, and actually they then match the HLA with the donor. If they found that there is a matching between the HLA between this recipient and donor, they perform the transplantation. And if they found that there is a huge difference between the HLA gene of the recipient and the donor, they do not perform the transplantation because the transplanted tissue or organ will be rejected. The second technique is called cross matching. In this case, they detect antibody against HLA of the donor that is present in the recipient. So if the recipient have any antibody against the HLA of the donor, they do not perform the transplantations. Now I will show you how this cross matching technique actually performed. This technique is called Luminex assay. In this assay, they actually take bead like structure that actually fluoresces. That means it has fluorochrome. This is called Luminex bead, and they use different fluorochrome for different beads. Let's assume this bead showing green fluorescence, then this bead can show violet fluorescence, and this bead can show red fluorescence, and they attach the HLA of the donor with this bead. So suppose this donor have 3 type of HLA. Let's suppose they attach the HLA with this Luminex bead. Let's suppose this is the HLA1, this is HLA3, HLA5. Abruptly, any HLA antigen. This is the donor HLA. Now they put antibody or serum of the recipient. Suppose if there is any antibody against any of these HLA antigen present in the recipient, then the antibody will bind with this HLA. Suppose this recipient have antibody against HLA3 antigen, then it will bind with this antigen. Now they put another thing into this system that is called secondary antibody, that is actually anti-IgG antibody. This secondary antibody can bind with Fc region of the recipient's antibody, Fc region. Now they tag this antibody with fluorescence. Let's assume this is the P tag 583. It could be PAR-CP or P-Psi7, any fluorochrome, but they tag this antibody with any fluorochrome. Let's suppose this is the P fluorochrome. So when this assay is performed, if there is any antibody against this HLA antigen, then this antibody will bind with this antigen, and they actually put another secondary antibody that can bind with the antibody of the recipient. So and they fluoresces, and they also fluoresces differently. So when this experiment or technique is performed, and if they found that the violet beads are actually fluorescing and associated with P tag, they actually come into conclusion that this recipient have anti-HLA3 antibody that is dangerous. This antibody can reject the transplantation. So this is called cross matching, in which they detect antibody of the HLA of the donor. The first experiment is tissue typing. In this case, direct HLA matching are performed. Okay, and in this case, antibody are actually detected. Now I will show you one way mixed lymphocyte reaction. Scientists take 2 different strain of mice. Let's suppose this is a strain X, any strain of mice, and strain Y. That mice are actually allogenic. Allogenic means genetically different. And after taking these mice, they isolate spleen or separate spleen and isolate lymphocyte. Same thing is performed in this mice also. Spleen are isolated and lymphocyte are separated from the spleen. Now let's suppose this strain X is the recipient, and if this is the donor, then one manipulation is performed into the donor mice, that is irradiate X-ray to this lymphocyte, or they actually add a chemical called mitomycin C. This X-ray irradiation to the lymphocyte or mitomycin addition actually inhibit the proliferation of this lymphocyte. Now you can understand why this is called one way, because in this case the recipient mice, the lymphocyte is called responder, that actually respond, and this lymphocyte, the donor lymphocyte, are called stimulator, because in this case recipient lymphocyte are not treated with any chemical or X-ray irradiation, that is responder. But in case of donor, the lymphocyte treated with in such a way that they do not proliferate. Now they actually put these two cells togetherly in a culture plate and also add another chemical, that is 3-hydrogen thymine or thymidine. Now they try to see what happens after this mixed lymphocyte reaction. They actually try to see the proliferation of the responder lymphocyte. Now I will tell you why these two lymphocytes are actually mixed and how from this proliferation scientists or doctor actually come into conclusion that the transplantation should be performed or not. Now in this mixed lymphocyte reaction also, scientists actually look the difference between the MHC molecule. What happens? I want to show you. This lymphocyte means specifically they try to see the recipient's T cells, how they proliferate in response of these donor mice. So this proliferation, actually they see the T cell proliferations, because this T cell is very important for the rejections. In my previous class I have told that CD4 positive and CD8 positive cells are important for rejections. These T cells that actually react, that actually helps to rejection, are called alloreactive T cells. Why they are called alloreactive T cells? Because they recognize this alloantigen. Now why they proliferate? The question is why? Why they proliferate? Why these cells proliferate? The answer is, you can see here the donor actually donating lymphocyte. They are putting this lymphocyte and this lymphocyte together. So that's why it is called mixed lymphocyte. We are mixing these two lymphocyte, mixed lymphocyte. Now we see the reaction. The response is proliferation. Okay, in this lymphocyte, this lymphocyte have T cell, B cell, dendritic cell. All these cells, these T cell, B cell and dendritic cells have MHC1 molecule, and this B cell and dendritic cell have MHC2 molecule also. Now these MHC molecules are the alloantigen for this recipient because they are allogenic. As they are different in genetically different, so this MHC molecule that is present in this donor strain, these responder cells actually see it as a alloantigen because this is different. They have a type of MHC molecule and this strain Y have different type of MHC molecule. When they come close to each other, as this lymphocyte cannot proliferate because we have treated these cells with X-ray or mitomycin C, so they cannot proliferate. Only this responder cell can proliferate. They are the stimulator cell. Okay. Now if they proliferate, as much as thymidine are incorporated by the responder cells, that means they are proliferating. Okay. Now how this proliferation actually tell the researcher that the transplantation should be performed or not? The greater the difference between MHC molecule of, or between this MHC molecule of recipient and the donor, the greater the proliferation occur. If there is a huge proliferation of these responder cells, that means there is huge difference between this recipient and the donor MHC molecule. So this is the indications. But here comes a very interesting questions into my mind, that how our T cells, I am calling it alloreactive T cells, are actually recognizing these MHC molecules, that is alloantigen. That means different, because in my previous class, T cell development class, I have mentioned they must agree with 3 terms and conditions. The first is they must self-tolerance. That means they should tolerate self-antigen. They should not produce autoimmunity. They should tolerate it. So self-tolerance of self-antigen, that is body's own antigen. Second term is lineage commitment. Each and every T cell must be CD4 positive cell or either CD4 or CD8. That means it is a T helper cell and cytotoxic T cells. And the third is that very important for this context, transplantation, that is self-MHC restriction. That means the T cell that actually develop into one's individual body always recognize the self-MHC molecule, only self-MHC molecule. This is called self-MHC restriction. The T cell can only recognize the individual's MHC molecule, not the other MHC molecule, because they are self-MHC restricted. Those do not recognize the self-MHC molecule, they die. Those recognize high with affinity with the self-antigen, they also die. So self-MHC restriction is important because those T cells are actually developing, they must agree with these three terms and conditions. Now I am telling you that there is some alloreactive T cells are present that can recognize different MHC molecule, alloantigen of different individual. That is very unusual. You have to think about it. So how this could be happen? Because these cells are self-MHC restricted. They can only recognize its own MHC molecule. So how this allo-MHC, I can call it this alloantigen MHC molecule, allogenic MHC molecule. I can give you a very simple experiment. Let's suppose this is the recipient and this is the donor. Now suppose this is a human being. If this recipient have a HLA1, HLA2 and HLA5. Suppose this recipient have these three MHC molecule, these three type of HLA molecule, and the donor have HLA3, HLA4 and HLA6. When organ transplantation or tissue transplantation actually performed, they actually reject. This recipient will reject because they have different antigen, different MHC molecule. These are alloantigen. But the recipient reject the donor by the help of these alloreactive T cells. These T cells can recognize these different MHC molecule, but these are the term that and condition that self-MHC restriction. They are all self-MHC restriction. They cannot recognize other MHC molecule. But how these alloreactive T cells recognizing different MHC molecule? The answer is allogenic MHC molecule that is associated with self-antigen are actually look very similar self-MHC with foreign antigen. The answer is when this MHC molecule are represented by dendritic cell or any endothelial cells, any cells of the donor tissue. The donor tissue have MHC molecule. It could be MHC1, MHC2. So when the donor tissue cell actually represent these allogenic MHC molecules, they represent with the self-antigen. The MHC molecule is not empty. They actually express self-antigen. Now our alloreactive T cells actually seem that this is the self-MHC molecule but with foreign antigen. So they recognize. Because the structure of the allogenic MHC with the self-antigen, when it is represented by this donor tissue cell, these alloreactive T cells actually seem or recognized as a self-MHC with foreign antigen, so they are actually activated and then proliferate because of this structural similarity of self-MHC and foreign antigen.